Wonderful. So, dzień dobry. Good morning, everyone. Thank you for joining us. We are officially recording, so we can start. Welcome to the second eye tracking workshop in the series. Today, we are talking about the basics of vision, eye movements, and measures, amongst a zillion other things. We have quite a lot to cover, so let me just briefly give you. And I think you need to be here. Perfect. To give you a brief overview of the plan for today, which is, first of all, to look at human vision. So we look at things such as the anatomy of the eye, the basic principles of vision. We look at parafoveal and foveal vision, as well as things like the parafoveal preview benefit and the perceptual span in reading. And then go into a bit more technical topics to do with the eye tracker, hardware and software, how it detects the eye and the movement of the eye. And then I will pass on the stage to Agnieszka to talk about eye movements and eye measures. Hi, welcome. So we'll talk about both the key eye movements and then derived, so primary and secondary measures, and we'll also cover early and late eye tracking measures. Then we will concentrate on calibration and validation. And here we really aim to give you as many practical tips as possible. We'll see a lot of screenshots from our lab where we have identified some issues with the tracking, so things to be aware of. And we'll try to give you some tips for troubleshooting. And then right at the end, if there is time, which according to my calculation, there will not be, we may talk and we may give you some additional practical research tips. But the first three areas are really the sort of meaty part of the presentation. So starting with the basics of vision, here I hope you can all see a picture of the eye. I just wanted to draw your attention on the key parts of the eye that we will need in order to understand how vision works. The cornea is the first one. This is the outer layer, right? This transparent lens that covers the eye. So typically when something gets into our eye, if we need to go to hospital because we cannot get rid of it through lacrimation, it is the cornea that will be potentially scratched, right? So it's the first outer layer. Then we have the iris, which is the colored part of the eye, the pupil, which is the central black part of the eye, more or less the center of the pupil. And this is basically the hole through which the light comes in. The light then goes onto the eye lens, which is located behind the iris. So let me see if I can use this. Wonderful. So the iris is the color part here, and this is the lens. This picture looks a bit misleading because the lens is actually behind the iris, and through this part of the eye, the image is focused and is projected back onto the retina. The retina is this layer, very thin layer that covers the back of the eye. I'm just going to use this because it's a bit easier. So right here, and it's a very important part of eye anatomy because here are located what's called photoreceptor cells. These cells are rods and cones. As you can see, there are a lot more rods than there are cones. Cones are responsible for sharp vision and for perceiving colors, whereas rods are responsible for night vision, for perceiving movement, and they are also responsive for what we perceive at the periphery, so peripheral vision. They are distributed differently on the retina. Hello, once again. And there is a, let me see if I can use this again. There is a very small spot right here. You can, like a little dip called the fovea. And in this particular area, there are literally no rods whatsoever. We have just a very high concentration of cones tightly packed together. And it is thanks to this small area that we can really see with sharp focus, so that we can have sharp vision. So here I just wanted to show you some more information about the shape of these cells. So rods, as the word itself suggests, a little bit like a stick, whereas cones are conical in shape. Surprise, surprise. Dzień dobry, welcome. As you can see, the cones can be divided into three types: red, blue, and green. If you wondered where the RGB sort of terminology that we have in photography comes from. We have these kind of light receptor cells right at the back of our eye in the retina, and the function of these is to perceive different frequencies in color. So the blue ones will perceive blue colors and the frequency in hertz associated with them. The main thing to say about these light receptors is that I quite like them because we work in translation, and you can call them the translator of the eye. What they do is they turn the light that comes into the pupil, they transform these light rays into electrical signal. An electrical signal is a language that the brain can better understand. And we will see that we don't just see with our eyes, but we indeed see a lot with our brains as well. So there's some operations that go on in there. So, but let's go in with order. So how does human vision work? How do we see objects? The light bounces off these objects or images or words, whatever we're looking at, and comes into our eye via the cornea. The cornea then will help direct the light towards the pupil and the iris, and these two parts of the eye work in conjunction with each other to adjust the amount of light that goes in, and they focus the eye onto the eye lens, and this is responsible for then projecting the image back to the eye, to the retina. Interestingly, the images are perceived upside down by the retina, and it is the job of the brain later, and we will see this, to then turn the image, flip the image back up. This is kind of similar to what happens with the pinhole camera, and the reason the image is upside down is that the hole through which the light comes in the case of human vision, the pupil, right, is very, very small compared to the real size of the object that we're looking. And this is without going into the physics of it, how the image becomes flipped. So the eye lens is responsible for flipping this image, then it projects back onto the retina, at which point the image is still flipped. So as you can see here, picture of a leaf, and the leaf is upside down once it goes on the fovea here. And then the magic happens, the light receptor cells do their job, the electrical signal then goes to the brain. So let's look at the brain a little bit more in detail. So once again, light comes through the pupil, goes through the eye's lens, onto the retina, then the cones and rods transform this into electric signal, very key aspect of human vision, the eye translators, these photoreceptor cells, and then the electric signal goes to the brain through the optic nerve, and the part of the brain that is responsible for vision and actively contributes to vision is called the vision center. It's located in the occipital cortex at the back of the brain. So the brain does sort of half the job, right? If we didn't, with the eyes alone, we cannot see. We also talked a little bit about the fovea. We saw this part. Let me just highlight it. So here you see a candle, no longer a leaf. And this candle, although you can't see super well, is right here. The fovea is the point of central focus. It is responsible for visual acuity. And why is this relevant to us? Because basically when we make eye movements, so when we, for example, read or look at any image, the very reason why we make these eye movements is to move the fovea so that we can see things in sharp focus continuously. So for example, when we move left to right to read text, these eye movements are necessary to have different portions of the text in focus so that we can identify the letters. Now, this is important for reading, but also for image processing. So the fovea, this area, is very small, about 2 degrees of the visual field, the very central part. Just around it, adjacent to it, there is the parafovea, which is 2 to 5 degrees of the visual field. And as you can, I hope, see in this image, although it's a bit small, you have here the fovea, then in the area immediately adjacent, vision is not as sharp, but there is definitely some information that can be perceived and can be used by the brain in interpreting information, including in reading. We'll talk about that in a second. And the rest is the periphery. So just here, I wanted to give you an example by saying, well, a few examples by saying that it's not only words, but it's anything that has a linear, so music scores, for example. And then with this screenshot here, I just wanted to make the point that there is a lot more periphery than there is focused vision. So inside the red circle here, we have foveal vision. Then between the red and the yellow, we would have parafoveal vision. And then all of this space is peripheral, and it can be divided into near, mid, and far periphery. And obviously, the amount of information that we can process diminishes as we get to the periphery. Now, a concept related to this and very important in reading research is the perceptual span. So if you're interested in working with language, particularly written language, this will be crucial to you using eye tracking. The perceptual span is basically the amount of information that can be captured at a given time, at a glance, and briefly held in working memory or in a sensory store. This is a fairly generic definition. When it comes to reading specifically, this area extends asymmetrically because we read from left to right. So it is wider to the right than it is to the left. So to the left, typically 3 to 4 characters, letter characters, and up to 14 on the right, at least in reading languages like English. And however, the word identification span, so this is the total span, the word identification span is actually a little bit shorter. Reading research has been able to identify that actually we can only identify words that fall within a smaller span of around 7 to 8 characters, again, to the right of the fixation. This is very relevant when we talk about parafoveal preview. This is nothing but having a valid preview of the upcoming word, being available in the paraphobia. This is very important because when we read, we do extract information about these upcoming words, even if we're not directly fixating on them. So for example, in the image that you see here, fox is the point of central vision, is where the fixation is what we're focusing on. However, we can still perceive a little bit of the word brown and a little bit of the word jumped. And this facilitates recognition later. What this means, facilitation benefit, is that basically fixations on these words that for which a preview was available tend to be shorter. Okay, we are faster at processing them. And sometimes, additionally, if there is enough parafoveal letter information that can be extracted with very small words, these can be perceived in their entirety. And therefore, the eye, with its sort of, might program its oculomotor movement to just go past these words entirely. So the words will be skipped. And we'll talk about skipping in a bit more detail later. Now, I want to stop here in terms of reading and in terms of how the eye works and what we know about the eye, and move back to looking at the eye tracker in itself, so the physical hardware and the physical software, and how this influences the detection of the gaze. Right, so this is a bit of a revision slide from our first workshop last month. So some of you may remember we talked about the PCCR method, the pupil corneal reflection. The corneal reflection is also called the glint, is this little blob. We'll see lots of glints today in the examples. It is nothing but the reflection of the light on this outer part of the eye, the cornea. And I'd like to mention dark and bright pupil detection. I find it quite interesting that actually traditionally detecting the pupil has been somewhat harder than tracking the glint, the reflection of the pupil. So there are different methods of tracking the pupil. Usually dark pupil detection algorithms are the ones that are most commonly employed. So if you are using an iLink or an SMI or other eye trackers, common eye trackers, you may have this. This type of detection algorithms works really well with high resolution cameras. So as recently high resolution cameras have become more readily available and cheaper, obviously, this has been the method that has established itself as the most common one. It is very good at dealing with gaze directions. So if we look at the 4 corners, which we do when we calibrate, we look at that later as well. These particular pupil detection algorithms are good at detecting the pupil, even when there is high rotation in the eyeball. So at these corners, where we typically might have some more issues in calibration. So they're quite good at that, but they're not as good in shadow and at perceiving elements that in infrared light, you may remember from last time, infrared light is bounced off your eyes by the eye tracker to track the eyes. So in these cases, however, there are things that are really dark that can and will be misidentified as the pupil, namely the eyelashes, for example, if there is a lot of makeup on them. So in these cases, it is the pupil detection algorithms that will not work. And we will see some examples of this, how the eye tracker misidentifies and just sort of marks with the same blue color these spots. So there will be some issues resulting from this that we will look at. Head movement compensation, again, I just wanted to remind people that even with the most modern eye tracking systems that allow quite comfortably to track people remotely, so we're not strapped in or we don't have any bite bars or any of these kind of historical eye tracking contraptions, but there is still what is called a head box. So an ideal, optimal range within which the eye tracker will track your eyes. If you step back, they cannot do miracles. I have to say it's quite good when you go back into tracking range at picking you up once again. But there is no miracle. You have to be still, you may have to adjust these distances to ensure that your participant is always visible. And then just to say that, the eyes can be tracked either monocularly or binocularly. Now, in a lot of the literature that we read is mostly monocular. The eye information is reported for one eye. However, you can collect data binocularly, and often you will have to, specifically, for example, if you're looking at microsaccades, then you generally collect data binocularly and you do some averaging. Two related concepts, accuracy and precision. We're probably all quite aware of what accuracy is, perhaps less so about precision. So let's look at both. Accuracy is defined as the average difference between the real stimuli position, which in our examples here is represented by the white dot, and the measured gaze position, so where the eye tracker thinks your gaze is. And precision, on the other hand, is defined as the ability of the eye tracker to reliably reproduce the same gaze point measurement. What does this mean? It means it relates to the ability of the eye tracker to group together quite confidently the individual data samples that form a fixation. This is very important to be aware of. When we replay our data, we see these fixations, but there are mathematical operations that go on behind the scene to group all these raw data samples into what then is classified as a fixation, right? So looking at examples of both of these, starting from the top left here, we can see good precision, right? Because these raw data points are clustered together quite well, okay, but fairly bad accuracy, because the position that where the eye tracker thinks the eye is quite far away from the target, so from the center here. Then we can see good accuracy and poor precision. So good accuracy because we're more or less on target, but the eye tracker isn't quite sure and the points are not clustered. Then perhaps the worst of the worst for accuracy and poor precision. So we're far away from target and the raw data samples are scattered. And then the holy grail, last but not least, what we are always trying to strive for with operations like calibrations, good accuracy and good precision. So we are on target, and however, also the eye tracker is quite confidently clustering all these points into the same fixation. So important to be aware of. Typically, we can directly affect accuracy, right, by changing position of the participant. We'll see different ways of doing that. Precision is something that more happens behind the scenes, but it's good to be aware of that. And this is all linked to the process of identifying fixations, right? We said there are these individual data points that need to be clustered together. So first of all, a definition: identifying fixation is basically translating these raw eye movement data points into individual fixations and their location, and by extension, the saccades between them on the visual display. This is usually done through event detection algorithms. And in the rest of this very long, apologies, quote, I just want to make everyone aware that it is still a subjective process. We don't necessarily see it. The eye tracker does it quite well behind the scenes. However, regardless of the precision of these event detection algorithms and regardless of their flexibility, there is still a subjective element to this process. We'll see in a second why. So there are two types of detection algorithms generally used: velocity-based, calculate the velocity of the eye based on successive data samples, and the unit of measurement here is degrees of the visual angle per second. And then we have dispersion-based algorithms, which instead look at a specific area, right, of the screen, the spatial box, and the raw data points must fit inside this area for a given period of time. So basically, velocity-based algorithms calculate fixations and then treat everything else that isn't a fixation as, saccade, and thank you, and treat everything else as a fixation, whereas dispersion-based algorithms do exactly the opposite. They identify fixations, and then whatever isn't a fixation is treated as a saccade. I will just say the velocity-based algorithms are the most widespread today. So all your iLinks, SMIs, and Tobii eye trackers will use this type. Yes, it's very good. It's a very good algorithm, but generally it requires high frequency eye trackers, so 200 to 250 hertz. So with old eye trackers, so if you have, for example, a Tobii, when I did my PhD, I did it on a Tobii TX120. This would have had either a mixture of both or just dispersion-based algorithms. But the modern eye trackers we use today, and certainly the Portable Duo, which we will use in the second semester, will use velocity-based. Again, not something that you will necessarily have to act on in your experimental design practice, but really good to be aware. And if you do want to know, you are entitled to ask the manufacturers. That was everything as far as sort of relations to the human vision and to the human eye was concerned. And we can move on to eye measures. So I'll just talk about the key eye movements, and then I'll pass on the word to Agnieszka. So major type of eye movements. Hi, hello, welcome. Major types of eye movements, we all know this probably, fixations and saccades. Fixations are movements when the eyes are, when the eyes are relatively still. And we say relatively because we will see later that there are some micro movements occurring within fixations themselves. They tend to be between 200 and 300 milliseconds when we're reading printed text. In the next slide, we have a bit more of a more precise average. Saccades are rapid eye movements. They're also sometimes called the jumps between the fixations. They tend to be between 10 and 20 and 100 milliseconds long, and they can be classified in forward or backward. Forward saccades in languages like English or Polish or Italian, or anyway in Europe, that when we read left to right, generally forward saccades means to the right, backward saccades mean to the left, and they are also, this last category is also called regressions. So a little bit more information about each of these movements. So fixation can range very, very widely. The individual variability is extremely high, from 50, 75 milliseconds all the way to 500 milliseconds and more. The average, this is the more precise sort of average I mentioned, 225 to 250, but always be mindful about this large individual variation. So you may find that your participants are on average, have longer fixations on average. That's fine, it is common. Why is this type of eye movement so important? Because it is used to determine a very large number of derived measures, which is what Agnieszka, such as dwell time, which is what Agnieszka will concentrate on. Saccades are on average 7 to 9 letter spaces long in English. They can range, so in total, from 1 letter space to up to 15, 20 letter spaces. So we can make saccades that are quite long. And some data by language. In a language like Chinese, typically saccades are on average 2 to 3 characters. In a language like Hebrew, for example, 5.5 character spaces. Regression, as we mentioned, are a subtype of saccades. They are saccades that move backwards in the text. They occur about 10 to 15% of the time in skilled readers. And what happens after this regressive saccade is that the saccade, the forward saccade after it, tend to be longer. And that's because we tend to go past the initial point from which the regression originated. And however, most regressions are to the immediately preceding words. Return sweeps are right to left saccades that join the end of one line to the next. So these are very relevant eye movements for us. We look at subtitles and how this type of text is processed. The subtitles are usually presented either as one line or as 2 lines. So when you have to make a decision in your experiment, how do I present this text, 1 line or 2 lines, be aware that whenever you have 2 lines, there will be these return sweeps. So oftentimes we tend to try and perhaps exclude the last and the first word in a line. You don't always have to, but you can do this. The first and last fixation on a given line are not right at the edge, so they're not like on the last letter of these words, but they tend to be 5 to 7 letter spaces from the end of the line. And within this space, about 80% of the text is typically perceived by these 2 extreme fixations. Skipping, we mentioned earlier briefly when we talked about parafoveal preview, why we skip words. Readers do skip words, and they do skip words of different types differently. So when we talk about content words, we tend to fixate on these quite a lot, right? Words that bear semantic information and that we need to understand what we are reading. These tend to be fixated about 85% of the time, whereas function words are fixated only 35% of the time. So they are skipped a lot more. And this is also because they tend to be short, so they can be fully perceived parafoveally and therefore skipped. Variables that really affect fixation and skipping are word length, number one. So as word length increases, the probability of fixating a word also increases. So we tend to look, the longer the word, the higher the probability of looking at it, and therefore the less skipping there will be. And with frequency works the other way around. So the more a word is, the more frequent a word is, the lowest the probability of fixating on it, and the higher the probability of skipping the word. Other types of eye movements, very briefly, and this is my last slide before I pass on to Agnieszka. Smooth pursuits. These are things that you will encounter every now and then in your eye tracking recording. We generally don't look at them, and instead of the eye moving in these jumps and then periods of relative stillness, which are the fixation, so rather than being hiccupy in this way, there tends to be a more coordinated and slow or steady velocity, a tracking of the eye. And this is generally applied when, for example, we are looking at moving objects. Now, text is generally still on screen, so we don't look at these eye movements. And then tremors, drifts, flicks, and microsaccades. All of these movements are sometimes called fixational eye movements because they occur within the fixation, right? So it's not just as simple as fixation and saccade, but within this stillness, there is movement and there are raw data points. So I'll just pass on now to Aga.
So, by the way, I just realized I haven't had my coffee. I left it in the kitchen.
Oh, no. But we'll some juice here if you like.
So let's see how that goes. Okay, so in our presentations, I think we are focusing largely on reading, and we want to stress that. So this will be focused on reading subtitles, reading text. But there's a whole lot of stuff out there on visual search and on perception generally that is not reading. Okay, just to illustrate and perhaps move on from what Valentina explained about different types of eye movements, we're moving on to how we can actually analyze this stuff. I'll try. Okay. Yeah. I need some coffee today. Yeah. Okay. So here we have a visualization of a scan path. Remember, we discussed scan path last time. So we have a visualization of how a person, a reader, is reading this sentence. So we have the sentence: "The knight attacked the windmill on his donkey." And you see that a person hesitated on the windmill because they were trying to process the sentence, and the knight attacking the windmill perhaps was not the first thing that you would expect here. So what happened? First, we had a series of saccades and fixations. So the numbers here, I can actually use this. Yeah. So we have first fixation, second fixation, third fixation, right? So and then saccades. So these are forward saccades, persons moving, and then they make the first fixation on the word windmill, another fixation, number 5 on this word, and then something's happening, right? They're processing, they're trying to understand, and then they go back. So they make a regression. So this backward saccade, right? And this this fixation number 6 is a result of this regression. So now they reread the word attack, they go back to windmill. So we can see that the word windmill, which by the way is our area of interest marked in yellow here, and in eye tracking, you would always have to tell the eye tracker what you're interested in, what your areas of interest are. Okay, so you have to mark them. We'll be analyzing words, but areas of interest can come in different shapes and sizes. So yeah, and they continue reading, making 10 fixations altogether, right? So what happened here on this area of interest, we can measure in different ways. Right, so we can have a look at the first fixation. That would be number 4, and that would be indicative of something. I have this explained on further slides, so just to show you. We can also analyze the first pass, so the first sort of round of gaze in this word, right? That would be fixations number 4 and 5 altogether. We can analyze the total time, that would mean the duration of all the fixations in this word, and that would mean we're interested in something else than when we look at the first fixation duration. And we can also be interested in regression path duration, which also includes the backward saccade here. Okay, so moving on from fixations and saccades, we're moving into different eye movement metrics. Sorry. You may be wondering whether we were wondering whether people read subtitles in the same way as they read printed text, and then generally the answer would be yes, although, for instance, the durations of fixations tend to be shorter. By the way, if you want to do some eye tracking research, it's quite important, and we learned the lessons here to make sure that you have the right distance between the lines. As you can see here on the left hand side, here the lines are quite close, so the result of such research, if we use the stimulus here, would be that sometimes it's not really possible to distinguish which line a person is reading. Okay, so whatever reading studies you'll be doing, you want to make sure that there is sufficient distance, and sufficient is also obviously a relative concept, but we need to make sure we don't just stick them together so that they're quite difficult to analyze later. All right, so common eye tracking measures. Okay, I have to keep clicking. Okay, all right, so these would be. Fixation count means basically the number of fixations that were made in a certain area of interest. First fixation duration is a measure that captures the duration of the first fixation on your area of interest, and we'll explain. Sorry, I'll just click through them all. Yes, so first fixation duration, and you can also measure mean fixation duration on this area of interest, taking into account all the fixations there. Time to first fixation, typically in visual studies, how much time it takes for your participants to notice something, to get into an area of interest. Dwell time gaze duration we'll explain in greater detail, right? You can also be interested in the length of the saccade. So in reading, people will be making short saccades, whereas when they're doing visual search, the saccades would tend to be much longer and then different directions, right? Another thing you may want to study is regression probability, or the probability of skipping, so skipping rates. Crossover, it's a term that we use in subtitling to denote the jumps between what we call the image area and the subtitle area. So whenever some areas of interest cross and you're measuring the saccades that cross between these areas of interest, these would be either saccadic shifts or crossovers. They're different terms as well. And finally, the pupil. So you've got pupil diameter or dilation. That's a measure of actually how your pupil reacts. We don't do that a lot because it's very sensitive to light, and when we use films, so the luminosity there in films from scene to scene differs, so that would actually be a confounding factor for us. So we're not experts here. Yes, so
it exists.
It does exist. We have to acknowledge that. Okay. Some people divide these eye tracking metrics into primary or secondary. So primary would be simply the ones that the eye tracker exports. You've got them right there, sort of very close to, I don't want to say raw data, but very close to raw data. And secondary measures, you'd have to do some mathematical calculations and operations to actually arrive at what you want to study. We have some examples, hopefully. Okay, so the primary measures would include, for instance, the number of fixations, right? They're just there. You don't need to calculate anything, the duration of the fixations, how many saccades you have, etc. With secondary measures, as I said, that requires some other measures as well, that you compare something with something, or you calculate stuff.
What's the difference between glance count and fixation?
Glances well, I'll get to that because that differs in terminology, but glances count would be the number of saccades made into an area of interest here. Yeah, let's perhaps go into detail into them, and we'll explain. So on the one hand, we had primary and secondary, and on the other hand, we have measures that are early, so happening at the very beginning. We're talking about, like, remember, like 200 milliseconds here, so it's still early, intermediate, and late. But early and late measures would what we would typically distinguish. So if you're interested in some processes that reflect these early stages of processing, you would want to use these early measures. And these reflect, as Val already mentioned, some properties of words. So word length and frequency and lexical status, whether it's a noun, a verb, function word, or a content word, etc. So you'll be able to detect how people already, with those early measures, with the first fixation duration, for instance, sort of see whether they can detect those things. And late measures happen, we usually take these measures not at the beginning, we sort of take the entire duration of the event that we are measuring there. So while the early measures sort of reflect the early processing and recognition processes, the late measures reflect how we're processing the syntactic structures or discourse level stuff. So more lexical and any other type of integration that is happening there, right? Or even we're trying to resolve some ambiguity, trying to understand this windmill being attacked there. That's where the late measure, I think, would come in useful. Okay, so what are these early measures? And what are the late measures here? So the early measures I already mentioned: first fixation duration, first pass reading time. If you remember the example with windmill, there were 2 fixations on the windmill, and then there was a regression. So in the first pass reading, we had 2 fixations, so we would add that up. And then skipping probability, again, could be defined skipping in the first pass, whether you skipped, or skipping entirely, whether you did not actually look at the word in its entirety at all. And late measures would include metrics such as total reading time. This typically is a sum of all the durations of the fixations, although some eye tracking manufacturers also add saccades to it. Rereading, so going back to an area of interest, fixation count, total fixation count, that would include all the different passes, the first pass, second pass, however many passes you had, that would be included here. Yes, and the measure that we haven't yet used, but we are very excited to start using is what some people call intermediate. So it's not a very early measure, it's not a late measure either, it's a sort of in-between measure, is the measure that is called go pass time or regression path duration, which would include the sum of all the fixation on an area of interest. So here the windmill example. So all the fixations here, number 4 and number 5, plus the regression that was made here before we move past the right-hand boundary of the word, okay? So if I went here and then went back, that wouldn't be included because I passed that on. So this is sort of first pass plus a regression here. Yes, so early, late, and intermediate measures. So now I have some slides basically on some individual metrics and basically tell you why you would want to use them or not. So we already mentioned first fixation duration. And again, I have this definition, so that reflects the duration of the first fixation. And we said already it's indicative of recognition and identification processes, and it is affected by the length and the frequency of a word, how complex it is, whether there is some polysemy there or not, some orthographic properties, whether you have to like go deeper or not, whether it's some something complex or rather more transparent. So if you're interested in that, use the first fixation duration. Mean fixation duration is slightly different. That is the average of all the duration of fixations on a particular area of interest. And typically, it is used to measure the so-called depth of processing. And I think a good example of this would be when you think about different types of text that we are reading. So if you're reading some light fiction, your mean fixation duration in English would be around 200 milliseconds, whereas you're reading more complex texts or text that probably we have all read in this department here, not physics and biology, but some more complex words. Yes, your mean fixation duration tends to be higher. Right, so that reflects some deeper cognitive processes there, and those people like Kalinka or Val or Carolina here might be interested in ERP as well. So longer mean fixation duration tends to correlate with larger amplitudes in N400. Now, dwell time is a problematic measure. Why are some of these measures problematic? Because depending on the eye tracker that you use, you know them by different names, and different manufacturers call them differently. Okay, so I'll just discuss this on an example of dwell time. So if you look at the definition of Eiling, one of the eye trackers we're going to use here, probably the best eye tracker currently, Eiling defines the dwell time as the sum of the duration across all fixations, right, in an area of interest. So we're adding up the durations of all the fixations. SMI, another eye tracker that we have that we're going to start with next semester, actually defines this in a similar way, but adds saccades. So all the fixations plus the time in between, plus the saccades. So in this view, obviously dwell time would be a larger measure, right? However, if you read one of the eye tracking Bibles by Kenneth Holmquist, he defines dwell time as one pass, right? So one visit in an area of interest from entry to exit. And if you go there again, that's another dwell time, okay? So you see, and then, so for him, what is dwell time for Eiling would be total dwell time and not just dwell time, okay? So you need to be very careful in what we do and what we use. And also when you're reading literature, is like you need to make sure what is it exactly that they used, which dwell time did they have in mind, because this is not always obvious. People unfortunately don't define things properly. Aga, you had a question? No.
Maybe I'll just jump in to say,
Sure.
to define your fixations when you write papers. I mean, we do these kind of workshops also
we have
to try and spread best practice.
Say that when writing the paper, you have to.
Yes, and not only describe your fixation, but also explain, if you can,
metrics,
why
yes.
are you using these metrics rather than other ones, right? Because as we have seen, we are seeing, there are quite a lot of them. So it is important to say, to make explicit the rationale why you are using this. Perhaps other people in the literature did it that you want to compare to, or these kind of things, or they are relevant to the construct that you want to investigate. For example, cognates, I'm looking at Agnieszka, you know, you may be perceiving them differently as soon as you first see them. So first fixation duration might be a good measure to look at there, and go past times as well. That's what I'm saying.
Yes, so maybe that's why we didn't, in our recent papers, we didn't use the term dwell time. We used total reading time,
I think it's clearer.
Yes, you can argue it's clear, but we also did define it, I hope. Yes, so that would include the duration of all the fixations, first pass, second pass, all the rereadings, whatsoever, the duration of all the fixations without the saccades for us, actually here. What does it mean if somebody has a higher dwell time or higher total time, total reading time? It might mean different things, and we don't know. We just have the number, and we have to interpret this number ourselves somehow. So it might mean that something is interesting, and you're looking longer for that. It might mean something's more difficult, more cognitively demanding, right? And you're having some difficulties in extracting this information. With the windmill example, that would probably reflect some higher level processing there, but could mean different things. Right. Another.
Sorry, those times differ from participant to participant. Obviously, they have to, right?
They do differ, yes. And as Valentina said, it's very participant dependent. So maybe you may want to. No, it's just idiosyncratic, I think. It's difficult to say. So maybe I don't know if I can make this jump, but maybe it's better to do like within subject designs rather than between subject design for this particular reason. Might be.
Yeah, I think there is a slide about this later, right? When we say that actually, you know, one participant will be quite consistent with its patterns, but these patterns may differ quite a lot between participants. So you might see that, you know, the average fixation for me is slightly longer than other people in my group. So instead of being 250 milliseconds, will be 300 milliseconds. But then, and I consistently make these fixations of this length, whereas someone else might be a lot quicker. So their fixations on average are only 150 milliseconds. So there will be these differences, and then you tend to be quite consistent within each other. But certainly individual variability factors are huge, and the field is turning towards them, right? Because things like age that you mentioned or multilingual. Absolutely, you have to collect a lot of information about your participants.
It doesn't necessarily translate that older people have longer fixations. It's not.
Yes, it depends on a host of factors. Yes, yes.
Yeah. Okay. But
the rate of unexpectedness, right? Like, you know, something appears there and it's totally unexpected.
Yes.
The fixation count.
Yes,
Yes,
so we looked at word length and word frequency, but this is by no means an exhaustive list. As you mentioned yourself, actually word predictability. So that certainly will play a role as well.
Definitely, yes. So if something is unpredictable, that would increase your fixation count, right? So fixation count, we said the number of fixations in an area of interest could be a word, could be a whole subtitle, could be a whole paragraph, could be anything else, right? And it's very much correlated with the total reading time. If you think about that, the more fixations you have, the longer time will be, right? So again, you can look at those things from different angles. Skipping probability, I sort of explained this already, right? So how likely is it that we are going to skip a word or an area of interest? Remember, it could be first pass skipping or total skipping. What we do, we usually use generalized mixed effects models for this because we treat this as a binary variable, so either skipped or not skipped, but I've seen also like percentages of skipping words or whatever, right? So that could go as a ratio as well. What else do I have? Rereading. It's a simple thing, although I did have a participant in London who asked me, what is rereading? So rereading is when you actually go back to a word that you read before. And again, we sort of can figure that. Backwards saccade, you make a backwards saccade to a word that you've seen before, you read it again, and yes, the windmill here, something is more difficult, you want to disambiguate stuff, etc. Okay, pupil diameter, PD, or dilation. As we admitted, we haven't worked a lot with this. When you work with it, you need to have a baseline. Okay, so you basically measure a change in pupil dilation against something. So if before you give people some stimuli, for instance, you might want to give them, I don't know, some just general text or whatever, something for them to look at, which would be your baseline, and then from this baseline you compare. This measure is tricky for us, I already explained, because of changes in luminosity in film, so we don't use that. But it also increases with the different state in which a participant is. So I would have a different pupil dilation if I had my coffee before speaking here today, right? But pupil diameter is very useful, and it reflects the cognitive effort, their the mental workload, as well as emotional arousal there. But you need to be careful with this. You need to pre-screen your participants to make sure they haven't got any conditions that are also quite likely to increase this. Want to add something?
Yeah, no, absolutely not. So pupil dilation.
Yes. Right, so I've already mentioned that. Okay, and this is what I said at the very beginning. There are huge differences in measures that you are interested, depending on what your study actually, right? Whether you study reading or visual scene perception. So the whole sequential reading, this what the sentence example that I showed with the donkey or the knight, sorry. It doesn't necessarily apply to watching videos or looking at images, right? The sequences would be different, right? People would tend to explore the image depending on, I don't know, whether their face is there or not, whether something is moving or not. So we're sort of focusing on reading here, but there's a whole world out there. And as I mentioned already, I think the duration of fixations tend to differ. We said that in reading around 200 to 150 milliseconds. In visual search, or when we look at people's faces, or the fixation durations are much longer. They go up to 500 milliseconds. Yes. Which again might reflect in reading greater difficulty of processing, and in visual search might just be an indication of interest, right? Okay, yes, this is about the mean fixation duration depending on the task. So we've been talking here about reading, but there are like different types of reading, really. Here FD stands for fixation duration. We'll just let's just perhaps look at this. So when you're reading silently, your mean fixation durations tend to be shorter compared to when you are doing oral reading, when you're reading out loud, right? So you're sort of listening to your own voice, and there's more going on when you're looking at scenes and in visual search again. So that the fixation duration, mean fixation duration, would differ. And the same with saccades, right? When you're exploring, saccades would be longer compared to when you're reading, and they're much shorter. Oh God, this is so boring, no? No.
The correlation of measures, yeah. So
Yes.
it's important though, because you will have to make some
yes.
changes.
We already mentioned this. So these measures are correlated. What does it mean? That means
the other increases.
yes,
Yeah.
that means there is a very much
with associated problems for the models.
Yes. So some people, like Conklin, for instance, she said that analyzing a range of measures to investigate the overall pattern should therefore be the aim, right? And if effect only emerges in one of your measures, then you should be really so if you're measuring if you're measuring something using 5 measures, right, and they should be correlated, but one is just showing something else. So she's saying basically maybe you shouldn't be really sure what's going on with this one unless there is a reason for it, right? So no.
Yeah.
No, do we have any?
Well, I mean, we can give you an example from one of the studies that we run recently, where we looked at skipping, we look at MFD, mean fixation duration, and we look at total fixation times, and something else that I can't remember. So, and we found an effect of, for example, we took away the sound, right? So we showed participants video with subtitles, and sometimes the sound was there, so there was this parallel processing that was going on, and some other times this was not the case. And we found that all not all 4 measures were affected by the presence or absence of sound. So whereas total reading time, right, would increase if the sound is not there, you really tend to spend longer, right, on those subtitle words. We did not find this for mean fixation duration and any differences between groups particularly. So native speakers and non-native speakers, although they have different total reading times with and without sound, they have comparable mean fixation durations. And then sort of the aim is to sort of know, what measures have been looked at already in the literature, what the measures are supposed to reflect, right? This is the key element. So there is still an assumption being made there. But so this is the example, right? We looked at 4 measures and one of them did not show a clear effect of our experimental manipulation, which in this case was taking away the sound. I hope that answered your question.
Yeah, so it could be something meaningful that is actually showing an effect, or it could actually alert you that maybe, just made a mistake somewhere on the way and they should be correlated, right? Like fixation count and total times, right? So
but sometimes it's interesting, right? Why does the total amount of fixation reflect this, right? But on average, so the average implies total fixation time divided by the number of fixation, then things are more or less equal, right, and similar. So we also looked at comprehension, for example, and if MFD, as Agnieszka said, is supposed to reflect depth of processing, paying more attention, potentially finding things more difficult, right? She made this example when we read different types of text, sort of a normal email versus very, very specific terminology to do with biology, we'll tend to make longer MFDs because those texts are harder, right? So if this is what MFD captures, then not finding an effect for MFD is very interesting, because it means that, of course, although our overall processing of the text changes in relation to having the sound or not, perhaps not having the sound doesn't impact so badly on difficult. So it's not that difficult for us to do, and then we found that the comprehension was not affected, right? So you use typically different fixation measures and different measures altogether. So for example, post-hoc tests, like if you want to measure learning or comprehension or recall, and then you look at these measures in conjunction with the eye tracking measure, and then you have a really powerful potentially design that can tell you a lot about processing. Sorry.
No, actually, I think you may want to say about.
Oh, yes, no, correct. So yeah, so the drawback of doing that, because then if we do this, then the temptation would be, well, let's analyze all the eye tracking measures that we possibly can, right? We've seen quite a lot of them. Well, that's not what we would recommend doing, and that's because whenever you run multiple analyses, you have to account for these multiple comparisons. Typically, your chance of making type 1 error rises, so you have to correct for this, and you quite harshly divide the p-value. So if we're looking at 4 eye tracking measures, we divide the p-value. Traditionally, is p-value that we use, the threshold is 0.05. We divide that by 4. So it means that for you to be able to declare this effect is significant, it cannot be 0.4. It has to be under 0.125, right? So much smaller, but you have to do this. So it's a balance, really.
I think we need to stress it's something new, right? It hasn't been done a lot previously. It's this new thing. It's this new trend, correcting for multiple comparisons. So if you read literature from, I don't know, 10 years ago, probably this wasn't done.
In statistics, it was done years ago.
Yeah, of
right?
course.
Yeah, in our field, yeah.
Yeah.
But I'm not.
Measures which are kind of.
But even in eye tracking books, I don't think, when you have a look, I don't think they recommend correcting for multiple comparisons.
I think Godfrey, it depends which,
Not all of
if
them,
you take Holmquist 2011, maybe he does not, but then if you look at Godfrey and other sort of more recently published manuals, that was published in 2020, you will definitely have something about that.
Yeah, so we'll talk about using all the possible measures in a moment. Here we just have the slide for something
we
that
basically said this already, so maybe we could move on, right?
We just basically explained
That's what your question, Agnieszka, was referring to. So as we said, if a participant might be quite consistent internally, but might differ to other participants quite a lot.
Yeah, so here we can detect a difference within subjects design, really. Okay, so the take-home message is here, though we're not going anywhere yet. First of all, we need to choose the right measure or measures. We tend to use more than one, right? But let's not use 10. So yeah, so if you want, for instance, in our case, to study how much time people spend on the subtitles, you may want to look into dwell time or total reading time. You can also calculate some relative things like proportional reading time against how much time the subtitle was displayed versus how much time people looked at it, right? You may, if you're interested in rereading, if you're using some difficult words in your subtitles, you want to study if people actually had to go back to your subtitles were too difficult, they had to reread them. So you would want to look into regressions and revisits and things like that. And our experience with replication, a very painful experience is this appeal to everyone that to facilitate replication of your studies, please provide, as we already said, the definitions of the metrics that you are using, and different companies use different names. Yeah, so here's just an example of a table from one of our papers, an old paper, where we basically say, okay, using some eye tracking metrics, okay, this is the definition, and why am I using it? Okay, it says longer mean fixation might be an indication of higher cognitive load, right? Absolute reading time is here, the sum of the duration of all the fixations, and Saccard, that was with SMI. Okay, so just provide a definition, and this will be very, very helpful for anyone who may want to study or repeat YOSTEM. And I wanted to show you an example of an eye tracking study I was reviewing, and the researcher provided, she was comparing three conditions: Polish voiceover, English subtitles, and Polish subtitles. And what she did is something that we are saying shouldn't really be done. So she exported all the data from the eye tracking software, and she put all the data in the publication or in the work that was. Okay, so all the different metrics that were there. I mean, there were more. I'm not showing them all here. And when you look at this table, it's sort of problematic in many different ways, right? For instance, if you look at area of interest, the dwell time on an area of interest, that's in milliseconds, but gaze duration is also in milliseconds, right? So it doesn't say it's in milliseconds for some reason. Okay, so obviously we need to make sure that we just give this information everywhere. And we can also see that she's using dwell time and gaze duration, and you can see they're very similar. Dwell time is a bit shorter than gaze duration, but we don't know which one's which really, yeah, because she hasn't provided any definitions. We also have something like glances count.
Can I have a question? Is it normal for studying with eye tracking to have such enormous standard deviation?
Yes. Yes, it's huge. Yes, yes, and that is quite common. Yes, I'm always baffled by this, to be honest.
Okay.
Yes, why do we have such a huge standard deviation?
Well, probably
yes,
because of the bell curve, right? With the Gaussian curve, you have quite a lot of because of this spread in individual variability that will.
Yeah, you would trim the fixations. What we do, what people tend to do, you would still trim. So trim mean you take the fixations that are no shorter than, let's say, 80 milliseconds and no longer
or
than
60
or
yeah.
60. It's just an arbitrary threshold. Some people end at 500, 800, 1200, right? So there is a limit there, but still, when you average that, and then across participants as well, with this variability, this is what you get, right? Yes. And then you have the glances counts and revisits, and you can see they're almost the same. They just differ by one number. We still don't know what that is. So perhaps I can now explain. So this researcher reported dwell time, and what did she say? Gaze duration, actually. And she was using SMI. So I went to the SMI manual to actually try and understand what she did. And for SMI in the manual, they say dwell time is the same as gaze duration, by the way. So she just, I don't know, somehow got the measure wrong, which is, as we said, sum of durations of all the fixations and saccades, okay? And the other measure that you may remember from this table, okay, here, so this one, the one that's slightly larger, okay, the second row here, is actually glance duration, not gaze duration. See, these words are very similar, right? So you just need to define them. It's like we're getting lost ourselves, right? And glance duration differs from dwell time in the sense that it also includes all the duration of all the fixations and all the saccades, plus the incoming saccade, okay? Therefore, it's higher, okay? So it's a tiny difference. I mean, did it make sense to use both measures? Probably not, right? Probably not. Just need to choose. The same with glances count, which is, and revisit. So glances count would be the number of glances, meaning saccades really, entering an area of interest, and revisits would be glances count -1. So it's not surprising when she reported her results, and this is a table for glances count, and another table for revisits, that when you look at these numbers, they are the same, right? Because this is sort of identical results here, right? This is identical to this, right? Because it's actually based on the same data. So this doesn't make sense really. This is something we shouldn't be doing. Jesus, yes. Let's move on. Okay, is it me or is it you?
You can just quickly go through it. I mean, or I can do it, no problem.
Yeah, it's one thing we want to say is, and I think we stressed that last time as well, is eye tracking in itself is interesting, but perhaps not sufficient. So it's always interesting to combine it with other measures. When studying subtitling, what we tend to use is comprehension, recall, cognitive load. We have some self-report questions. We tend to measure people's immersion and their immersive tendency, enjoyment, and different things, whether they manage to recognize a subtitle or a scene, etc. And then some participant-related things like their working memory capacity and
language ability as well. So proficiency, for example, if you're working with native speakers versus non-native speakers.
Yes, their familiarity with subtitling.
There's a host of individual measures that really come into play, as Agnieszka asked about earlier, and we do recommend always combining more than one eye tracking measure, and combining eye tracking data as a whole with other methods of data collection, and triangulate your data in this. I think this is as much as we want to. We can.
Kind of, if go back to this for a moment, because some of these measures will be related to participants, okay, and some of them will provide additional dependent variables like MRI, because you kind of listed them.
Yeah, this is just all together. Yes, they are. They absolutely are, right? So sometimes you could create correlations. For example, Agnieszka earlier mentioned that some correlation between ERPs and some of the fixation data. So you might run correlation analysis on this. They will be or individual independent variables that you add to models as well. So if we can then move on, because we don't have that much time, and I just wanted to spend a little bit more time talking about practical things. So now we got all the theory out of the way. We talked about all these countless eye movements and measures. I'd like to concentrate on calibration, right? So practically speaking, in order for the eye tracker to detect this host of amazing variables that can tell us a lot about how we move the eyes and what we do when we process words and images, we need to calibrate the eye tracker. This calibration is a procedure that is required in video-based eye tracking. And what it does is basically creates a map. So maps what? Features detected in the eye image. What are these features? The glint, as we saw earlier in that slide on gaze detection, and the distance from the center of the pupil. We talked about this last time in the first workshop. So mapping these features detected in the eye, that's why we have the video camera, that's why they're called video-based eye trackers. So we detect these features, and then we map these to the position of the gaze on our display screen. right? So in order to do this, typically you have some calibration points, they are called. Here, I wanted to show you this. This is a really old screenshot from my PhD. This is from the Tobii eye tracker. Very, very basic. We will see much more complex screens very soon. But this is as basic as you can get. Here we have 5-point calibration procedure. Basically, this is a process where we ask the participant to follow this dot that moves on screen randomly. It will stop in many different places. And the task of the participant is simply to follow the dot as closely as possible and to fixate always in the middle of it. After you've run this procedure, you may encounter some problems. So here, bottom corner or top corner problems. So typically related to the fact that actually the rotation of the eyeball is highest when we're looking at things that are at the periphery of our area of our screen area of interest. So here, for example, we can see that we have a slight bottom corner problem. So these green lines that come out of the fixation target are the errors. This is a very, very coarse a way of and you don't ever get any numbers, so all you have is this sort of visual way of assessing error. With iLink, which we will be using in our practical, there are much more precise ways of establishing whether your calibration model is correct or not. So anyway, when you have these kind of issues, you may have to do some troubleshooting, and we'll look at quite a few examples of that. And then I would like to mention also that calibration is, at least in iLink, iLink does record these values, right, and the error, which is calculated in degrees of the visual angle, and so you can use this information at a later stage when you want to assess the quality of your recordings. Naturally, some participants will be tracked better than others, and you may have to exclude some participants due to eye tracking data quality issues, while calibration values, if are available in your eye tracker, are one way that you can assess whether the model, the initial model, was accurate and precise or not. And if it was, this still doesn't mean, you know, I can have a perfect calibration and then fall asleep halfway through the trial, so you won't see my eyes. So this is not automatically related to having high quality data, but it's possible that if the calibration model is already bad, then chances are the quality of your data will need to be examined more closely. So here, just to show you, this is the setup that we will be working with. This is the Portable Duo. So in front of the screen here, we have the display PC. So this is where the participant will take the experiment. This is where we show them films. This is the Portable Duo eye tracker here, and you can finally see the infrared illuminators, several of them. This is how the light bounces off your eye and comes back to the screen. And this instead is the host PC. So this is typically where the eyes would be, right eye and left eye. And in this window here, we'll be able to see the participant's face. Obviously, I'm taking a picture of this whole setup now, so you cannot see my eyes. Yeah, so you can see your hand taking the picture. And so this is one setup, and I wanted to show you this because this is exactly what you will be getting your hands dirty on if you come to the practicals. However, we do have another, this is Agnieszka taking our experiment. We do have another setup. This is the 1000 plus in a room that's just behind the corridor, just behind us along this corridor. And here you can see that Agnieszka is in the chin rest, right? And all of these elements can be adjusted. So you can change the height, and you can even slide the chin in this. So this rest pad slides, which I'm mentioning right now because it will be coming handy later when we talk about troubleshooting some issues. This right here is the eye tracker, which is in fixed position. We recommend not to move it. What you can move is the distance between the participant and the height, so the angle at which participants look at the display screen. And here we have the experimental station where we would sit, and you can see the eyes clearly, right eye and left eye. In previous, in my experience as working with these systems, I didn't always have the eye, and I have to say that once you start working with iLink and you start seeing the eye, it's like buying a smartphone. Okay? You didn't have it before, but you're not going to go back really to not having Google Maps and Google Translate, right? So once you work with this, you will really see and appreciate the benefit of seeing the eye, because at all times during data collection, you can check live, so you can make some notes. For example, we have a lab book, right, that allows us to track if anything goes wrong, if the participant fall asleep, for example, right? If you're not behind them, you cannot see this, but you can through this. So some information about it. Now we have Sonia here, so lots of examples. Here we're working with the Portable Duo. We are in remote tracking mode, as we can see here. And I just wanted to draw your attention to the optimal distance. So as you can see, the eyes are visible on screen. This is the left eye and this is the right eye, right? And there is an optimal distance. We talked about the tracking box earlier, right? The trackable range. And this is where you would typically look for the optimal distance. Now, Sonia, our participant here, is in an optimal place indeed. You can see this little dot here. She is.
And Sonia has this dot on her.
Yes, this is the remote sticker, which is here, and you can see it also on her head. This is what enables the remote tracking mode, right? So it is quite outstanding, actually, the amount of movement, both laterally and. Of course, there is a range, right? So you don't want to get out of that box, but within that range, the eye tracker is actually very good at tracking you. And then very important, I cannot stress this enough, the crosshairs. So here they're a bit small, so we will see lots of examples of them. Yes, it's a sticker. So you get given a role, right? And then one participant after the other, you give the sticker to them, they apply it on, or you apply it.
Yeah, if you can do this remotely, then you would have to send the sticker to this person, right? If you want to.
So if you say remotely in the sense that you want to use web-based eye tracking, that's something else. You don't need the target sticker there because what you use is the so you're limited anyway to the capabilities of the computer that you are the person that is doing. So during the pandemic, for example, this was a wonderful alternative to not being able to do research at all. But there are some major limitations still in these types of
remote.
Yes, so by remote, we don't mean remoting as in you are at home taking the experiment. We mean remote tracking. So you don't need to be strapped in to be fixed, but you are, you can move your head, right? So Sonia here is able to free her head, to move her head freely because there is the third eye, as we like to call it. And
kind of easier for the participant. It's more like
yes, it's the most ecologically valid, right? Because you're not stuck in this chin rest.
One more, sorry. So if the participant, for some reason, like looks away, and you lose the eye tracking for a moment, will you get it back?
Yes, absolutely. Now, this is a good question that you do because because when you are tracking participants with this system that we see on screen now, actually, if you are doing this, if you go way too far, right, then the eye tracker makes a horrible noise, and it happened to me once. So just the participant was, maybe stressed because we tend to, to move when we watch something. So within limits, right? But yes, so the eye tracker will lose you and make a horrible noise in this particular case. The iLink doesn't 1000 plus does not do that, so you just lose the eyes, right? But that's why it's so important to have the picture of the eyes because you can monitor this, right? And then absolutely, the eye tracker, as I think I mentioned earlier, is quite good. As soon as you get back within trackable range, your eyes will appear in these two areas here and here. And I wanted to show you this. I'm going to quiz you a little bit. So can you tell me what is going wrong here? Either people in the chat, feel free to unmute yourself, or people in the room. What do you think? What can you see? Yes, exactly. So the eyes are simply not there. You can see pupil missing here. And the reason for that is they are way too far in this case. So you can see the dot is in the red section. So all that you do in order to correct this is sit the participant closer. Or move the eye tracking station closer to them if you can do that. I would just also like to draw your attention to this line of text that always is at the top, right? So if you, for example, forgot whether you've switched cameras or whether you have, what kind of the tracking mode you're in, you can always check this. This information is telling us here that we are in remote mode, that we're collecting data binocularly using a 25mm lens. And we are tracking at 500 hertz. So although the capabilities of this eye tracker are quite much higher than that, you can indeed collect data at 1000 hertz. In this case, we are halving that speed and we are collecting samples every 500, at 500 hertz. Then a few more examples. So here again, the crosshairs. I even included myself just to be as harsh in the pictures. So here we can see and I bet you didn't notice that this is me. Yeah, so I shouldn't have said that. Okay, I'll keep that in mind. Just don't mention it and then no one will notice. So the crosshairs, again, are noticeable and they have been identified perfectly. And the pupil has been filled with blue. So this is good news. However, generally, there is also a line right, that is drawn around the pupil. So this means that the pupil detection algorithm that we mentioned earlier has worked correctly, right? The pupil has been identified, which is half of the job. The glint has been identified, so we can be sure that the eye tracker will have fairly sure that it will have high level of confidence in tracking the eye. Now, can I quiz you once again and ask you, to look at the picture on the left and to tell me what might be going on there? So particularly looking here first, where the arrow is on the left, what do you see? Yes, someone in the audience here said reflection. There is a reflection. It's called glare, this reflection. It comes from the fact the participant is wearing glasses, right? So as we can see here. And so what we see is that there's two turquoise blobs, not one, right? One is the correct one, is the glint. So in the left eye, we can see that the crosshairs, this cross is correctly on the glint, like it was here. However, for this participant, things are a bit idiosyncratic. So its glint is not at the edge of the pupil as it is here, and it is most of the time, but it's kind of inside the eye, right? And what do we see in this very dangerous position, which is on the edge of the pupil? We see an extraneous glare for the glasses that is right there where the glint usually is, all right? So here, actually, the problem is I can very see this as problematic, but there is no problem. But if you skip to the next eye, then look at what the eye tracker is doing. The eye tracker here, the pupil detection algorithm, the glint detection algorithm is getting it wrong, right? So the glint is the smaller dot that's inside the pupil that's totally disregarded by the eye tracker, which instead is using this larger blob, right? This larger reflection coming from the glasses, and is mistaking that as the glint. So all of this in pre-calibration, you look at your participants, and this is where you make a lot of your fine-grained changes. You might change the angle. You remember I mentioned this, the pad that you can slide backward and forwards. You can change the height. I will talk about all of that. But this is in order to correct for this, right? So ideally, you want no glare. In practice.
I have a question. So what will happen if we don't correct that?
If you don't correct that, it's possible that the eye tracker will use this glare, right? So and will basically be less precise in the calculations of where your gaze is, because you need both measurements to be accurate. You need the pupil because you need to find the center of the pupil, and then you use the distance between the center of the pupil and the glint, right? So if this distance, if we're using this particular glare here, then the distance is no longer the real one, right? So this will mean at the very least that your gaze detection algorithm will be less precise and less accurate probably as well, if you can track at all, or the eye might be lost on a higher percentage of the trial. I also wanted to draw your attention to the fact that, remember earlier we talked about pupil detection algorithms. We said that the dark detection algorithms are the most widespread, and we said that they're really good when looking at corners, but not so much when it comes to elements that may be misinterpreted because they have the same color in infrared light than the pupil. So you see the pupil here is marked in blue. However, this part, so the eyelashes are also marked in blue. Now here, this is not a huge problem due to the eye anatomy of this particular participant. So where we see that the eye, there's quite a lot of iris, right? So I'll just use my mouse. So right here, I hope that you can see the mouse. There's quite a lot of iris in between the pupil and the eyelid. But in, for example, participants from Oriental areas of the world, they tend to have an eyelid that is a bit lower, right? So in these cases, if the eyelid almost touches the pupil, well, then these two blue areas are very close to each other.
Some people just tend to squint a lot.
Yes, and I'll talk about squinting in more detail in my next few slides. But if these two are too close, then the pupil detection algorithm won't work. It will think that the eyelashes are part of the pupil, and this is going to disrupt your tracking as well. Just quickly, so this particular participant is not wearing any glasses, and yet we have some interesting clusters of extra glares and reflections, right? Turquoise in color as marked by the arrow that may be misinterpreted as the glint. However, as we are seeing, the glint is correctly picked up. The crosshairs are on the right turquoise blob, but be aware when there are other blobs there as they may interfere. Same thing here, so just different examples. So in these cases, you may want to readjust distances and angles and to see if you can can get rid of these glares, right? Sometimes if they are there, they are really big and square and they are far away from the eye, right? Glasses, then they won't matter at all. It's just when they are small and really close to the pupil that they can be misinterpreted as the glint. And then I want to ask you again, what is going on here? What do you think in this participant, in this particular case? What is the problem, do you think? So the pupil is being detected, right? It is filled in with blue and there is a round circle around it. Can anyone sort of guess, maybe in the chat as well, unmute yourselves, sorry, in remotely unmute yourselves and try to diagnose, give me some diagnostics of what might be going on here. Yes, there it looks at why that might be, because actually, if you look, I mean, I try to. So first of all, one eye is working okay, right? The glint is picked up, and there is crosshair. So the problem is only with this eye. Why? And there is a blob here, right? So what is the? Well, it might be. We don't know. Why don't we know yet? What do we need to do in order to see better? This is. I'm giving you a hint. So I'll tell you. So there is such thing as focusing the camera lens, and there are rings on both eye trackers, including the one you will use, where you have to, just like the eyes need to have clear focused vision, so does the camera of the eye tracker. So here, and you typically can do this eye by eye, right? So in this eye, obviously, the camera is in focus and we can find both the pupil and the glint. Here, as we can see, it's much blurrier, right? So, but the glint is already there. It's already found the pupil. So the detection algorithm actually works in suboptimal conditions here. But in order to find that glint, you need to focus the camera simply, right? So sometimes, it's something that don't work, and then you realize, oh, hang on, I just need to go and turn the focus on the focus ring, right? And then again, pre-calibration. So these participants tracked really well. However, just to point out here on the left-hand side, we can see that even if I am fully within my trackable range, I'm looking ahead, the moment I move my eyes even to the right or to the left, so sideways compared to our plane of interest where we're supposed to be looking at, then obviously the eyes are there. And actually the pupil detection algorithm, as you can see, still trying to find the pupil and to fill it with blue, but there's no chance of finding the glint in these cases, right? So you just have to tell the participant, because this happens sometimes. People want to talk to you, so they might, after you've put them in place, you've calibrated, you're ready to start the experiment, they have a question, and what they do is they come out of the chin rest and they ask, right? So and then you have to redo the calibration all over again.
Or they don't, they just try to move their eye.
Well, yes, I mean, so they try to, so they try to be helpful, so they stay where you put them, so where tracking is optimal. So just say, I just mentioned this to them generally, you are allowed to ask me any questions, just do it by looking straight at the blank screen ahead of you, which is a bit strange, but there you go, it prevents problems like those. Then Sonia here mentioned squinting, laughing also does achieves a similar result. So obviously the space, your eyes become smaller, right? And so this can create issues between the pupil and the eyelashes. However, here I'd like to point out, this participant is laughing. I asked him to do that so I could take the picture. Look how well the detection algorithms still are working, right? So we cannot see the top and the bottom of the iris, but the pupil is still there. The crosshairs are there. Tracking can happen without any issues. So this is just little praise to the latest systems. If you didn't have the eyes, you couldn't look at any of this. So I hope you are starting to realize how useful it is for troubleshooting issues, and therefore for increasing data quality in your participants, and ultimately to make sure that you can use the data from more participants, so there is less attrition in the sense of data that you need to discard later on. Here I just wanted to point towards some interesting borderline case. Oh, we have 2 minutes left. Okay, so here I'm showing you this because it's borderline, right? So you can see, so that the participant is watching the clip, that's why you can hear the audio. You can see how the glint sometimes is there, sometimes it's not. The pupil sometimes is detected, sometimes it's not, right? So
sorry.
I'm going to stop because we don't have that much time. It's just to say sometimes there will be borderline cases like this, where here you can see, for example, that only the left eye was tracked. So usually for this experiment, we collect data binocularly. So this is already telling me that we have so many issues with the right eye that we decide to track only one eye. This is one way of getting around the problem. Is the data from this participant useful? I mean, this is the kind of troubleshooting that you can do whilst the participant is taking your experiment. However, later on, I would mark this participant as worthwhile checking the recordings, right? And then you'll have to make a decision.
Do you think it's because of some illness they have? Or
It's difficult to say, really. We do collect information about if they have eye conditions beforehand. You can screen your participants in this way. Generally, glasses and lenses do not constitute a problem in and of itself, but if in certain cases they can, right? And then so if you do consider these elements beforehand, you might be able to, you know, if a participant tells you straight away, I have this eye condition, and then it turns out that the eye tracker contact lenses, we'll talk about that. So first of all, I just wanted to very quickly show you, this is a real example of how the pupil detection algorithms takes it. I hope you can see it. There is a green line that is not exactly on the pupil. It includes, basically, sorry, we have a light reflection right now, so that's not helpful. But you should be able to see that the basically the eyelid is included in this count erroneously. Here, the illumination is the main problem in this screenshot on the right? So we can see the pupil. We can even detect the glint, but there's no crosshairs, right? The light, the illumination is too low. So troubleshooting calibration. So first the issues, and then I'll give you some solutions.
Maybe
Illumination problems. So I'll just take, we'll have just 25 minutes instead of 30 minutes for questions, if you don't mind. This is
we're about to finish.
Yeah.
oh, the time's over. Okay, well, I'm so sorry to. So you can ask questions if you like. I would love to talk about this.
Yeah, we'll talk about it.
Yes, we'll talk about it next time. In particular, I'll just leave this slide on the screen to say that there are solutions for all of these issues. And when you come, if you come to take the practical classes, we will get you to calibrate, recalibrate, and deal with all these issues. So
I would add this, like, talk to your participants.
oh yeah, instructions. Yeah, participants' instructions are really key, both beforehand and when you come to the lab as well, right? So anyway, time is up. So we'll stop sharing the screen, perhaps,
And stop the recording.
and stop the recording. Thank you very much for listening. We hope that it has been useful, and you're interested in learning more and in taking some of the practicals where you get to use all of this technology and to see for yourself when it fails and how to troubleshoot effectively.